Dear students, now that you are aware of scoring matrices, we would like to see how many types of scoring matrices exist. Now you may ask, how come scoring matrices are of different types? So the answer to that will be that based on our former concept of the frequency of occurring of certain amino acids within biological sequences, it may be necessary to understand that there are multiple strategies to compute this frequency. So towards that, we will try to understand the first type of scoring matrices, that is the PAM scoring matrices. The alignments are scored very nicely if we use an empirical scoring matrix. That is a scoring matrix based on experimentally observed frequencies of amino acids. And as I just mentioned, there are multiple types. For instance, there is PAM matrix, there is Blossom matrix, and there are several others as well. So to start with, let's take a look at the PAM scoring matrix. PAM is essentially point accepted mutations matrix, and it means that for a given site in a protein, the probability of one amino acid being substituted is 1%. Or simply put, the point accepted mutation is a substitution of one amino acid by another such that the function of the protein stays the same. Note that there can be a case where one specific amino acid in a protein is substituted and the protein function changes. So we are not talking about such a case. Let me reiterate the purpose of PAM. So for PAM, we are only considering those mutations or substitutions by which the function of the overall function of a protein stays conserved or stays the same. So towards that, we need to understand what is the PAM unit. Simply put, the PAM unit or the point accepted mutation unit is a time in which about 1% of the amino acids in a sequence goes under mutation. So that is called PAM1. So you must understand that since the sequences are long and there are multiple nucleotides or amino acids in the sequences, so when I say 1% of the sequence is changed, does not necessarily mean that 100% PAM will have 100% variation in the sequence, because the same site can be changed more than one time. So if the same site is being mutated multiple times, then the entire sequence may not be changed for the entire protein. So to understand that, here I have a graph here for you, so in which the sequence difference and PAM distance is compared. So if we are naive, then we can think that a PAM distance of 100 will mean a 100% change in the sequence. But that is not the case, because one site upon mutation may further mutate and thereby get and accumulate multiple mutations. So therefore, the experimental data shows that if you have 100 PAM distance, only 55 to 60% of the sites in a protein are actually mutated. So if you grow further, then the PAM distance may increase to over 300 for an 85% variation in the sequence. So to compute the PAM matrices, so the first thing that you need to do is align the protein sequences which are one PAM unit diverged, that is, they have about 1% mutations. Aij is the number of times Ai is substituted by Aj, that is, one amino acid Ai is substituted by another amino acid Aj, and you count the number of times, and also you calculate the frequency of each amino acid Ai. You simply plug these values here. Here K is equal to 20. So there are 20 amino acids. So you sum the product of frequencies for these and you obtain PAM 1. And if you want to use PAM 2, then you just square this. Similarly, if you want to use PAM N, then you have to multiply PAM 1 by itself n times. And this is what you get if you multiply PAM by itself 250 times, and you get the substitution matrix like this. So in conclusion, PAM matrices are scoring matrices that are useful in comparing and scoring the protein sequences. You can compute PAM 1, PAM 2 and so on till PAM 250 and even more. And PAM 120 is considered to be the best choice for comparing similar protein sequences.